there is a reason behind that. Thank you so much, for your kind invitation, and also thank Dr. GPS Raghava. So I've been asked, to. I hope you can see the screen and my voice is audible, right? Yeah. Yeah, thank you. So, this session is related to multi-omics. So, over the last 10 years, ever since I've been into mainstream postdoctoral works, I've been working on integrative multi-omics approach. The last talk by Debashish was a wonderful setting because I started my career as a proteome researcher, where I worked on hypothetical proteins and slowly drifted towards, next generation sequencing over the last 10 years. So we're all, in the realm of these COVID times, and I think, we all need to collaborate, converge, and try to remain consistent. That's where, we can probably, combat this COVID crisis. So this is what, I call it as the 3 C's. But the midst of this, we have a wonderful word called machine learning that's been going rounds over the last 3 decades, more over the last 3-4 years. So I usually keep this particular cartoon, as you could see, this wonderful, inscription from the pebble stones, that this girl who is really lost in an island, she says that, she wants help. She collects some pebble stones and writes help. No one comes to her rescue. But she realizes that, yes, she's a machine learner, and she senses by saying that, I know machine learning, and even, kind of a jet aircraft, comes to rescue. So my talk, would largely delve into some of the SNP prioritization approaches, largely based on the annotation strategies that we have been working. And albeit the fact that we have developed certain pipelines, if not the tools, these particular pipelines have not been integrated into the web CGI, but I'll just come to that particular thing. So these are my sections for the talk today. I'll slightly move towards some of the teasers, the problems and challenges for variant classification, the transgression story, which I'll slightly delve upon, and some of the case studies knowingly of prostate cancer. So this paper came up yesterday in the Lancet, which is quite alarming, and they also emphasize that when they have collected such huge data related to anxiety disorders from 204 countries, certainly they used multi-omics approach. But here, the most important thing is not just the multi-omics approach, but also the quantification. Taking the know-how of these particular data is also absolutely, very, very important. So if you really see, this is primarily to all the PhD fellows. I have 6 PhD fellows, and I can sense, how the last, 19 months has been. We always wondered, is COVID-19 a blessing in disguise or a kind of a mere despair? So I think, I'm sure, every one of us have been piggybacking between, these 2 syndromes, the cognitive bias, the imposter syndrome, and the Dunning-Kruger syndrome. So on March 22nd, to be precise, when the lockdown was announced, I think, we had, we were at some point of time, mounting, towards a kind of a stupidity over here, where, we knew that, we knew everything. That's where, probably, that would happen. And by the time, summer would come, maybe, SARS-CoV-2 would deplete, and that was what, we were all thinking. But, it was the other way around. We are still, coping up with COVID-19 fatalities. And in March 2021, it was a big havoc. There is absolutely, a despair here. So we are so-called impostures, even till now. But I'm sure, we will probably, become experts not just in the COVID-19 research, but also, tackling, the COVID-19. So this is where, probably, we are in. And if we can, fight this particular picture, I'm sure, we can become, wonderful researchers. And that would also allow us to teach wonderful, work-life balance. So this is what I was just talking. So all my probably scripts and the pipelines, all of pipelines are put up in this GitHub or Bitbucket Prash bio. One of these particular prioritization things, for non-synonymous mutations, we are trying to put up in HIPO2, which is not yet done. And we started a consortium called CAPSE, which is a Cancer Prostate Consortium of India. I think some of those particular prioritization methods are put up through this. So this is the only thing, and these are the two important pipelines that we have developed over the last 3 years. So I come from Amrita University. I recently moved here last September. The last 5 years I was at Birla Institute of Scientific Research in Jaipur. We have a lush green campus, as you could see, wonderful backwaters, merging into sea. We have got a brackish water somewhere there, merging into sea. Wonderful in atmosphere, but it always rains. Even now it's raining. It's been about 44 days I've been here and haven't seen, the rain stopped. And the good thing is ours is an institute of eminence. We have got about 34 faculty, all coming from the West. And ours is among the top, 10 institutes in India and top 100 in the world. So when it comes to the health, as a systems biologist, I've always wondered, that bioinformatics has always moved up the value chain. If we have taken any 3 verticals of the central dogma of bioinformatics, like the sequence, the structure, and maybe the functional relationships, you have at some point of time, you would always talk about the functional reporter, but the strong basis for this particular functional reporter is the mutations. So you take any organism, you ask, what are those particular mutations, that are probably specific to them, which probably are not are absent in the control population, and we are still asking what are those particular mutations. Sorry, what are those particular mutations? What are those particular strains that are really adapting and unfitting in the environment? The best is the SARS-CoV-2. So systems biology or the systems genomics approach, taking the realm of multi-omics, has really moved up the value chain, and it was once lost into oblivion. And now we are always talking about several entities. We're always talking what specific mutational spectrum can be seen even at the stage of single cell genomics or single cell transcriptomic data. We are not only talking about the differential express genes, but we're also talking about those mutations that are carving out from those particular differential express genes as well. So this is a kind of a wonderful discipline, but in between this, prioritizing a mutation, taking the GWAS data, taking the whole exome data, whole genome sequencing data, is always a challenge, because I started my career working on some complex diseases, taking the diabetes data, taking the rare disease population, where we wouldn't even find mutations that are probably common across the subpopulation. So we would ask whether or not that particular mutations would really fit into the current diseased phenotype of interest. So then we have these wonderful guidelines. The American College of Medical Genetics and Genomics, the ACMG Guidelines, the Association for Molecular Pathology, the UK-based consortium called Association for Clinical Genomic Science, and it's also associated with the Australian agencies. So they have given wonderful prioritization. They have given best practices and guidelines, and yet we always needed to rely upon how good the data is, how are we really talking about the quality data coming from the whole exome, whole genome integration, and when we are working on some rare diseases, and it's always more challenging because we need to help the patient. We need to ensure that yes, we need to come up with the challenges of taking that particular patient data for viable genetic testing. So there are, of course, some evidence-based classification strategies, which are promulgated from the ACMG guidelines. So if you find a mutation or a SNP of interest, probably we could classify based on whether or not it's strong or very strong or supportive evidence. And of course, now the most important thing here is whether or not a SNP is pathogenic in nature or likely pathogenic. And these days, we're always talking about the variants of unknown significance or variants of uncertain significance as well. So given such large amount of data, we need to come up with less discordance, or we need to come up with a concordant set of mutations. That's where is certainly the kind of bottom line. And of course, when we run the NGS pipelines, when we do those particular things, we sail through all these particular databases, like DBSNP, the DBVAR for structural variants, and of course, CleanVAR is the clinically relevant pathogenic variants. Of course, there are other proprietary databases like Genomade, Genome in Bottle, the CAD, and the GERP. All these particular scores are also tabulated, and then we prioritize the variants. And of course, we have the wonderful databases from Vinod Skari and Samir Brahmachari's labs, and also Sridhar Sivasubbu's lab, like Indigen Consortium, where we also take into these particular things. And of course, these particular data are fetched from wide amount of data, the whole genome sequencing, the whole exome, the target genome sequencing, and all those kind of things. As I was telling you, there are certain mutations of interest, and of course, at some point of time, we would also be very much interested to look into the variants of unknown significance. So I just try to give you a kind of an example, why prioritization plays a very, very important role. You'll be surprised to know that there are 5 important DNA repair genes, like these 5 that are listed. I'm sorry, the BRCA1, BRCA2, MLH1, MSH2, and CFTR, largely at the interface of this mismatch repair DNA system, where there are 50 missense mutations in each of them, which brings a kind of a challenge. How many of these mutations are really pathogenic in nature, for a diseased condition, for a diseased phenotype? To check that, people have tried employing, lots of, panels. They develop some kits, and of course, well known the whole exome, kits, right from the Agilent, the Twist kits. They have all these particular things, that could capture anywhere between 34 MB to even 68 MB of whole exome data, okay, across the human genome. So because 1.8% of the human genome is coding, so that amounts to about 56 MB of data, constituting the exomes. Then we would ask, what happens to the non-coding SNPs? Because the genetic variation is best seen in the non-coding RNAs. So if you are doing whole exome, maybe we may not really find non-coding SNPs, but if you're doing whole genome sequencing, if you need to catapult interest, if you have some monies, then we can certainly identify the non-coding SNPs. And how do we prioritize them? So these are the kind of things. And when we sit with clinicians, we always need to brainstorm. First we come up with certain variants of interest. We classify them. The clinician would say probably, taking our disease condition, maybe, I might expect this gene. So, hey, look, do you find any mutations, coming from these particular genes? And then we might come up with some concordant functional assays. We reconfirm. We might also ask, if we really need to identify any, mitochondrial variants, per se. So, moving forward, when the next generation sequencing approaches have really come up, now it's no longer next generation sequencing. We have these wonderful, capture-based approaches like the DGS, the direct genome selection, hybrid capture-based methods, the PCR, of course. There are some, approaches, taking into, consideration. But for a young audience here, how these particular mutations can be taken up at the end, I'll just try to give you. So imagine somewhere you are in Delhi, you're paragliding somewhere 10 kilometers above the sea level, which is about 10,000 meters. You cannot see anything, excepting that you might think that yes, you are somewhere in Delhi or NCR or somewhere. But when you come down to about 5 kilometers above sea level, you can clearly see the sky of Delhi, maybe you jot down to about 2 kilometers above sea level, you come up and see precisely that you can see India Gate or maybe JNU or somewhere, those particular things. And then, that's where, you call it as the depth of the coverage. And that's where, you can identify what kind of interest it is. Is it, you're talking about the data that is what you really need to obtain from an exome population, which amounts to about 6 GB in case of a whole exome data, or if it is a whole genome sequencing data where you have to shell out a lot of 24 GB to 240 GB data for one run for a 30x coverage or 40x coverage. So you need to ideally understand from which source you are trying to lift these mutations, identify those particular SNPs. Okay, and of course, whole transcriptome shotgun sequencing, you don't talk about what you call the depth of the coverage, that you talk about the mappable reads, or the transcripts per million reads. In case of a paid-in data, you talk about the fragments per kilobases per million reads. So these plays a very, very important role. And of course, whether or not you are using a reference population or a de novo population, your mutational spectrum also varies. So what we did was, this was a problem, that I've been working, with my group members. This was a problem that we encountered as a part of my postdoc in 2013 and 14. There are two important terms in genetics called pleiotropy and linkage disequilibrium. Pleiotropy is nothing but not necessarily one gene is accorded with only one diseased phenotype. So you might have one gene that is associated with several diseased conditions. But you have got a linkage disequilibrium where in a small chromosomal loci like this, you have got several SNPs, and you fail to identify which of these particular SNPs is a pathogenic one. So if you can identify from those particular small, say 2 kb regions, say you get almost about 650 SNPs, you identified some 650 SNPs, so which means 1 in 3 SNPs tend to be associated to be pathogenic or likely pathogenic. Then you're asked a kind of a problem. You ask with several questions. Would all of these particular SNPs in that particular region tend to be pathogenic in nature? Then how do we prioritize? So this was a kind of a problem when we were working on the bovine data. This is a data between in chromosome 6 between SLC4A4, some small region of SLC4A4, and MPFFR2. There's also a GC gene somewhere sitting here in chromosome 6. In chromosome 4 in humans, if you take the analogy in humans, it's in chromosome 4, and that's where the vitamin D gene is associated. The GC gene is associated. So this was a particular problem, but we had that particular data was coming from the genotyping population, and we had the whole genome sequencing data as well. And now out of those 650 SNPs from a 2 kb region, which of them is pathogenic? That was a problem. And of course, as someone who was interested in hypothetical proteins or the known unknown regions, I was also largely interested in the long non-coding RNAs. So we developed a kind of a strategy. We developed a kind of an annotation approach, and that we didn't publish until 2018. We were not confident. And we used different machine learning based approaches. We did some prioritization of SNPs. We had to manually look into those particular regions, walk through the genomes, 5' UTR, go through the ECR or the electronic, what do you call the 5' UTR regulatory regions, the open chromatin elements, the OCR, and all those kind of things. And then, we had to manually check them, which I'll try to, tell you in the next slide. And then, we would call those particular SNPs as the causal SNPs. We wouldn't call them as bona fide SNPs, because out of those 650 SNPs, we need to jot down to the list. Okay. So this is how it looks. So this is a small region of this. If you look into a genome browser, like an ensemble genome browser somewhere here. So this is chromosome 6 as indicated here. And if you zoom in, you find a lot of SNPs here. And we really don't know what are those particular SNPs. Are we really filtering by criteria by looking into the depth? Or are we really looking into the genotyping population? Or are we really looking into certain data that is available from the concordance and all those kind of things? So we came up with a three point classification strategy called the functional annotation. So we would look into the presence of signal peptides, the transcription factor binding sites, and the shared pathways. So if a SNP is there, if within the SNP, the vicinity of that particular SNP, do we find any of those particular enriched motifs? And likewise, those enriched motifs, are those a part of the evolutionary conserved regions? So which means if those particular SNPs are a part of the GURB scores, that we would really look into this. And of course, we would also see for any of those particular SNPs are a part of those enhancers or the promoter regions, or even the non-coding RNAs, because that's where the genetic variation, albeit the fact that it's best seen in non-coding regions, you might find a SNP that is present in the non-coding RNA, which tends to be highly pathogenic in nature. So these are the things. So we have given some scores, for every SNP that is falling in these particular criteria, and this is what the score lies. So I just put it in a more pictorial here. So imagine, these four are different features. Of course, there's also another fourth feature, which we have largely taken into consideration for non-coding RNAs. So imagine these circles, small circles, are all the SNPs, So now this guy, the SNP is lying in all the four features that we have identified, whilst annotation. Okay, so we have the SNP here, we have the SNP here, we have the SNP here, we have the SNP here. Now, the simplest thing is we consider that particular SNP to be highly pathogenic or diseased in nature. Of course, we don't end up there. We prioritize those particular SNPs, we check the pathways, and then we, of course, run through those, precision and recall-based machine learning integration. We run through at least 2 or 3 machine learning integration based pathways and come up with a sensitivity. That is what we have done, and we have published that annotation strategy. We haven't developed what we call the web CGI for that. We are still working on that. Once we gain confidence. So the part 1 of my talk, the conclusion says, we have prioritized SNPs of variance using a 3-point classification feature system. That would probably belie taking the annotated records from the heterogeneous data sets, taking the functional annotation, the enhancer elements, the methylation, probably the pathways, and even the spectrum of the non-coding SNPs. You might ask why and where these particular SNPs prioritization approach would be helpful. That is where my next 2 parts would really be discussed. So when I moved back to India, I was doing almost 7.5 years of postdoc. I was piggybacking. I was running out of ideas. I had several thoughts, but when we came back to India, we thought that I'll work with prostate cancer because that caught my interest. So we thought no one has really worked on prostate cancer, the mutations relevant to India. So that's because prostate is for those aged men above 55 or 52 who are really prone to the enlargement of the prostate gland, and because of scrotal pain or whatever, the person gets uncontrollable pain. And there are 2 variants. One is the benign prostatic hyperplasia, which is not so malignant kind of tumor, but the other one is a malignant tumor with a Gleason score of above 5, some pathological specimen. So we also have some other characteristic features which I'll not discuss. So whenever a clinical condition is obtained, we look into the prostate serum antigen. We also look into the the Gleason scores. We also look into the distal rectal examination reports and all those kind of things. But in India, it has been alarming. So we thought, we should probably look into those kind of things. And then we asked whether the SNP prioritization approach that we applied could be applicable in this particular approach, because anyways, it's a SNP prioritization approach. So we had a pilot project even before BrainPan and Novartis funded us. So what we did was we came up with a very pilot study with taking 10 samples, because getting samples is also very difficult task. And then this COVID has happened. So as you could see here, all the square boxes are all those particular genes that have been inferred from whole exome sequencing approach as a diagnostic front, where there are about 30 genes that are causal to prostate cancer, and these squared, genes are largely seen in Caucasian or the West. So when we have done the whole exome sequencing for Indian population, we would probably anticipate that some of them would be here. Of course, we did get some of the OTOF, we get some of them associated with the obesity as well, some of them with vitamin D as well. But these circles are Indian specific variants or the genes. But here, as you could see, we knew that BRCA1 and BRCA2 are largely associated with prostate, but from Indian spectrum, we found a lot of variants that are associated with both BRCA1 and BRCA2. Okay, so BRCA1 is something exceptional, So this is something interesting. So because this is where, had we not used those prioritization approaches, we would not have gotten these particular SNPs. So this is where our SNP prioritization based approach was really helpful, and this was what the Gleason scores that were largely helpful. And you might ask me why Prash, why prostate cancer, why is that you have started with this particular thing. Usually prostate cancer is caused in a lot of those people who eat red meat, largely in Africans and African Americans or American population, or Caucasian, or the so-called the European or whatever. But in India, about 73 samples that we have collected so far, you'll be surprised that largely from Jaipur, 69 are vegetarians, from our informed consent. Okay, only 4 are non-vegetarians, and yet they are prostate cancer victims. So we are trying to ask, there must be some diet related things, there must be some microbiota, or there might be some diet related genes underpinning these particular mutations. This is one of the reasons why we asked. So the part 2 of my conclusion says we have applied those particular SNP prioritization based approaches, taking or identifying those particular mutations that are largely associated with prostate cancer in Indian subpopulation. So we found all these several mutations, which I'll not go into details, it's on the slide. So we have developed this conceptual framework of mutations. So whenever we prioritize the whole exome sequencing, right from FASTQ reads till the VCF or the bed file that is created. So we'll ask our PhD fellows, don't look into those particular SNPs as pathogenic alone. Now come up with our approach, walk through the genomes and see if any of those particular SNPs run through those 3-point classification based approach. And now what next? So as I told you from my first slide, we should always talk about the machine learning based approaches. So this is where we are talking right now. So this is slightly different. I'm slightly moving forward. So when we were going through this thing, we got that particular paperwork published. A paper came up saying that the prostate cancer victims our prostate cancer patients are more likely to survive from COVID than the normal. That caught our interest. And then, this is where we try to achieve this, which I'll try to come up and say. We found some long non-coding RNAs that are largely, associated with this. So what we are trying to do at this point of time is we have some whole exome sequencing data from which we have called the variants, taking our SNP prioritization approach. We have used a part 2 from the public available databases where we have used the RNA-seq datasets. Of course, we're also running our own datasets as well. And then we are trying to use a deep variant, which is a machine learning based approach, taking the variants from the whole exome sequencing data and the whole exome and the data from these, the SNPs, from these particular data, and now come up with a viable training module and ask which of these particular variants are really pathogenic. That's where, we might come up with prioritizing those, 24,000, variants, to a normal shell. So the third part, which we recently published last week, I just thought is slightly different, but this is also a so-called a prioritization approach. When the COVID-19 came in, although I've been, piggybacking between those Imposture and Dunning-Kruger, we have been fortunate enough to have published about 10 papers, wonderful papers, very satisfactory papers, in COVID, thanks to the wonderful collaborators. So we asked, what are those epitope targets across those particular underlying ORFs because I was largely interested on the hypothetical proteins. So there are known unknown regions in the SARS-CoV-2 as well from the non-structural proteins, because there are 4 structural proteins like the membrane envelope nucleocapsid and the spike. So so what happens is, this virus, this little big virus, is really turning very intelligent, because today we have got the so-called Delta variant, and there's a news that even the Delta variant recovered patients couldn't catch up the herd immunity. Even if at all if we are to take up, even a city like Delhi couldn't take that. We are asking why. The simplest question is why. So the essence of this particular story is, I'll go back to this particular slide, I'm sorry. So we have ACE2 and TMPRSS2 as 2 important receptors over here. And this is SARS-CoV-2 entering the host. And these 2 are the receptors which receive SARS-CoV-2 at the entry. And no sooner it receives, the mitochondria gets hijacked. This is what we have published. And as a result, the cell undergoes asphyxiation. So if there are about 2,000 mitochondria per cell, all of them die, and as a result, the cell dies, and as a result, the tissue dies, and as a result, the person dies. That's a story. In milieu, there are some long non-coding RNAs, sitting somewhere over here. This is what we hypothesize. They keep on watching, what this ACE2 and TMPRSS2, how they, act as receptors for SARS-CoV-2. And they try to interact with these 2 guys, and of course, along with some interleukins and interferons, present in the cell as well. So this is what we have simply done using a simplest sequence-based approach, which we call it as a transgression approach. Okay. So there is a non-structural protein NSP15 here, which is present in SARS-CoV-2, which transgresses the long non-coding RNA, which is sitting in the host, and this long non-coding RNA is non-HSAT. Okay, and this long non-coding RNA is absolutely taking up again some of the bases from catenin, which is a particular protein coding gene present in the host. So which means the NSP15 link RNA, the long non-coding RNA, non-HSAT, this ID, and the catenin harbors the same amount of bases all through. So which means there is a transgression. So whenever an infection takes place, this long non-coding RNA chips in, but the viral protein is integrated with this long non-coding RNA, and it takes the exact basis of this particular host, protein or host, gene as well. So this is what we call it as, the transgression story, or a trespassing story, and that is probably, heralding how. What happens to the genetic reported in the host. Then you might ask me, what's the story behind this? So we have also prioritized our SNP-based approach. We're looking into these particular data. How different are the variants coming from this, and this? So this is what we are trying to attempt, so that we can reproduce our works, so that whatever editorial that we have published last week, we can make it live. And of course, our works are also underway in CCMB for metatranscriptome analysis of this SARS-CoV-2. So this is what we have published. So SARS-CoV-2 transgressing long non-coding RNAs uncovers the known unknown regions in the human genome. So the overall conclusions from our slide, from our works is, of course, prioritization plays a very important role, not just in the adage of technical or scientific interpretation of calling the variants, but also for the economic advantage. So if we can prioritize more number of SNPs, and if we can come up with some validation study for few of them, then that would really save our time, for Sanger validation and other stuff. And of course, I'm sorry for this typos in this thing. So we also had those linkage disequilibrium, which I was talking about. So such prioritized variants can help identify those things. And of course, we don't simply prioritize variants and take up those variants from our walking story, but we also take up the statistical and functional approaches. And that's where we can bring up the confidence scores. So the bottom line here is prioritization helps us achieve bona fide SNPs. So the so-called the causal steps, that's where we can take up. I am a co-editor for this Frontiers in Genetics. We have a call here, so I would like to request if anyone has a very important paper, we can give a full waiver for this integrated systems genomic approaches for characterizing uncharacterized proteins. So you're most welcome to contact me for this. And I'm a founder of BioClues. Over the last 17 years, we have been mentoring several graduate students. We have mentored so far about 3,000 graduate students of about 5,000 strength. Many of us students are largely placed across 30 countries. We give them, we provide them, we teach them the ethics. GPS Raghava was our first president, and we have published over 55 publications. We try to reproduce the data. We teach them, some basic etiquette of reproducibility as well. We have several virtual projects, and thanks to wonderful mentors. And this time, we are working. We are organizing INBEX, the Indian Conference on Bioinformatics, during November 11-13 at CSR Neast in Jorhat. So you're most welcome to give your submit your work. Last time, Vinay Panda was also there, as a speaker. So with this, I think my time is up. I thank GPS Raghava, Shipra, and the chair, for the wonderful invitation. This work would not have been enough, without these wonderful group members. They have been good listeners. They have been, most of them have been imposters and Dunning-Kruger's as well. They have been absolutely amazing PhD fellows. Nidhi Shukla worked on those transgression works along with Harshita. Purva and Sonal, they were my postdocs. They worked on those prioritization approaches, which we tried to, reproduce. And of course, thanks to my wonderful collaborators and mentors, and thanks to my funders. And of course, I've recently joined this, and thanks to our Dean, Bipin Nair, and the World Humanitarian Amma Amritanandamayi. Yeah. I look forward for the questions and brainstorming session. Thank you so much again. Yeah. Thank you, Jaspreet. Yeah.